Hello. So, one thing about doing a PhD in the UK, or at least where I am in the Quantum Biology Doctoral Training Center at the University of Surrey, is that the way things are set up is during most normal times, you can get a good night's sleep every night if you want to. So, this week, I decided to push my limits and see what I could get away with. So, I did a sleep experiment where I went to bed an hour later than I normally do because I usually have a pretty consistent schedule. But what I didn't realize is that doing so, I ended up getting around 6 hours of sleep each night, which is suboptimal for me, and I get quite affected by it, by loss of sleep. And also, what I didn't realize is that this week we would start doing lab work in the lab very consistently every single day. And 2 days this week, we worked late hours, and those same days until 7 after 7 p.m. And those same days, I was at in the office and or the lab for an 11-hour workday each day on suboptimal sleep. And this is because we were doing a PCR and an assembly and also a separate protein purification, which I was still happy to do. I enjoyed shadowing and learning and doing what I could. But I can tell you this information because one thing I also did was keep track of the hours I spent in the lab, just roughly to within 5 minute sort of margin of error. I tried to keep track of when I went and left and what I counted as sort of being there or not. And I think that helped me realize how much work I was doing, which is good. It was 26 hours this week in the lab, and that doesn't count like lunch breaks, for example. So that's like time actually spent in the lab and not in the office. And also, I think it's useful to keep track of the time you spend doing certain things if they can be driven by sort of obsessive tendencies. Like, for example, one night I stayed and made plates, even though had just finished something that I might have screwed up and I volunteered to do in the first place. After I'd finished that, I said, "Well, I'm waiting for this to incubate. I might as well make some kanamycin plates." And right now I'm still learning, but once I am more competent and in complete control of the time I spend in the lab, I want to be aware of how much time I'm spending in the lab for my sanity. So I know how much I'm actually working, and I can kind of evaluate, well, is this okay? How am I feeling based on how much I'm working? How much can I really handle is what I want to learn so that I can work at optimal efficiency and work the maximum number of hours at the optimal working level without decreasing the quality of work, because we're talking about scientific research. If I'm mentally absent like I was many of the times this week since I was on suboptimal sleep, then it's not even worth doing it because you will fail 9 times out of 10 with when you're cognitively if you're significantly cognitively impaired, and the lack of focus slows you down anyway. Because I have ADHD and bipolar, which both of those mental illnesses make you sensitive to things like lack of sleep and can result in significant impairment to your cognitive function. But the upside is that I got to spend a lot of time with Alejandro, my student mentor, this week before he leaves I'm not sure. I know he's not sure when, but he's finishing soon, and within a month won't be able to spend any time in the lab together anymore. But I don't know if he'll leave or stay depending on his next steps. But it's been good working with him. He gives me wisdom, tips, and tricks for protocols that he does, like inverting the orientation of centrifuge tubes when doing multiple centrifuges of a sample. But he also told me that staying late is important sometimes because staying late can help you save like 3 days worth of work if you do it right. And I think that having obsessive tendencies can be helpful for this type of work because, for example, to really understand PCR at the molecular level, you need to have a curiosity for detail and be able to keep track of what you understand and what you don't, and sort of visualization helps too. And then also doing things over again, because that will happen. It has happened, will continue to happen. It's part of the necessary effort and process of research and just developing samples in biological work, molecular biology specifically. And compulsively writing down what you're doing is also very useful when it's sort of a compulsion, when every time you do something you feel like you need to record it. That can be helpful in the long run, these sorts of traits. And even if it's you learn over time. Even if you do it too much at first, you learn to dial it back, or you can learn. It's harder to maybe force yourself to do it if you not used to doing it. But there are loads of different techniques to getting the lab. One drawback of ADHD, though, I'd say that is that inattentiveness is terrible. It's very challenging because there was a time when I don't know if it was this week, but I started to do something and I just didn't spray down the lab bench, and I was like, and I didn't notice until I finished. I was doing sterile technique and I just didn't spray anything down, and that's something you might want to write down if it's for an important procedure, things like that. But Alejandro, being towards the end of his PhD, has also told me that he calculated that every publication you get saves you 25,000 words of writing on your thesis, approximately, which is some encouragement. And I've also received some interesting advice today that once you start learning about your topic, your subject, you should start writing for your confirmation, which is what they do here instead of a qualifying exam. The confirmation is essentially an update after your first year of your PhD. But I've just finished my first month of my PhD, and I can say that I think it's going well, and it's definitely increased in busyness, but I think that's overall for the best. But I still feel like I have control over my time, and things are manageable and pleasant and not incredibly stressful or anything like that. So that's just sort of where things are at. More work in the lab, and it's only going to increase over time. But it's satisfying. It's good for my dopamine levels working in the lab, putting in effort, and I feel like I am suited to this work. I wasn't sure how it was going to go, but I think I like it. And I look forward to continuing next week. I'm going to do what I shadowed this week. I'm going to do it myself next week. So, next week I will let you know how that goes. But for now, thanks for watching, and I'll see you next week.